thank you for coming and accepting this invitation, and we are hoping to hear from you.
Yeah, thanks a lot, Anna. Thanks to everyone for coming to my talk. And yeah, thanks for the invitation. Yeah, really, this is my first time here at CREAF, believe it or not. Yeah, and don't ask me why. We are living this crazy life we live, and I didn't have the chance to come. And yeah, this is my first time here, and it's really a pleasure to be here. As Anna said, I'm a professor at Rey Juan Carlos University, albeit next year I hopefully will move back home to Alicante. And I would like to present in my talk today some of the research we are doing on the ecology of drylands. This is, as Anna said, I come from Alicante, so I have a personal attachment to this type of ecosystems, and these are the ecology in general of terrestrial dryland ecosystems, and how they are responding to ongoing global environmental change are the main foci of our research, of the research in my lab. So this is the typical slide that I present when I'm giving a talk in Sweden, France, or some places with more temperate climates, that many people really don't have they all believe that drylands are like the Sahara Desert and systems like that. You all know how drylands look like. We are talking about ecosystems where there is a limitation of resource, mostly water. So as a result, we don't have a continuous vegetation cover. We have this typical distribution, no matter if you are in Tunisia or in Kenya or in Almeria or in Peru. You typically have this discrete or this mosaic vegetation where you have discrete plant patches surrounded by an open matrix, which traditionally in dryland ecology has been always talked about a bare ground, but as I will show you later, these parts without perennial vegetation are really important for the functioning of these ecosystems. So drylands are really very important ecosystems worldwide. So the latest estimates indicate that they cover over 45% of the terrestrial surface, and they are the home to over 40% of the global population. So they are really important ecosystems, both from the economical, sociological point of view, and also very important from the ecological point of view. And drylands are important today, but are going to be even more important in the future. If we follow and we believe the latest forecast from the IPCC and the latest simulation models, they predicted that by the end of this century, the global extent of dryland area will increase between 11 and 23%, depending on the emission scenario that we consider. So drylands are very important today and are going to be even more important in the future, tomorrow. But when looking at the ecological literature on many important topics like biodiversity, ecosystem functioning, about biogeographical studies, most of the literature on key ecological topics over the last decades have been carried out in ecosystems other than drylands. There is really a very important gap in many aspects of our understanding of the ecology of terrestrial ecosystems that we need to cover. We really need to pay much more attention to drylands, because as I say, they are really important because of the extent they occupy, and they are crucial also for the sustainability of our planet, because most of the human populations with the largest demographical increase are located in dryland areas, which also will suffer a lot because of forecasted increases in aridity conditions. So what we are trying to do in my lab is we try to understand how dryland ecosystems function, how they are responding to ongoing global environmental change, and we do this using a multidisciplinary approach, multi-organism, multi-scale approach. We use all the tools we have available to fulfill this task. So we work at different spatial scales. We work from local scale studies, usually carried out in drylands here in Spain, mostly in the Madrid area. Also we have sites in Almeria and Alicante. We also work at the regional scale, the Mediterranean, in Patagonia, in other areas, and we also work at the global scale. Usually we conduct manipulative field experiments at the local scale, and more observational studies, remote sensing, using also remote sensing information at the global scale. We are also doing more and more mathematical modeling, and we are also doing a lot of work in the lab. We do a lot of biochemistry work, and we are doing more and more molecular work, mostly working with soil microbes. We are working with plants, vascular plants. We also work a lot with organisms forming biological soil soil crust, which I will present them later, are these communities dominated by mosses, lichens, and many other microorganisms that are really abundant in drylands worldwide. And we are also, as I say, working more and more with soil microbes, with bacteria, with fungi, archaea, and also with protists, and we are starting to work with nematodes too. So I would like to present some of the work that we have been carried out, we have been doing in the last years at the global scale, and then at the end of my talk I will present some of the work at the local scale that we are using to confirm some of the patterns that we are doing or that we are observing at the global scale. So this work has been carried out thanks to a global survey, the EPES Biocom survey, that I have been coordinating and we have been doing over the last 10 years or so. We started this when I came back to Spain in 2005, and this has been, we are still processing data from this survey, but this survey lasted for over 10 years. And this is a very interesting survey because it's the first global standardized field survey that has been carried out focusing on drylands. So we were able to survey thanks to, this is a collaborative effort with more than 60 scientists involved from 30 research groups and universities in more than 20 countries all over the world, except in Antarctica. And so we were able to survey over 230 site drylands from 21 countries. And in every country, in every place in the field, we characterized the vegetation using a standardized protocol. We collected soil samples, and all the soil samples were shipped to Spain, where we analyzed all of them either in our lab or in other colleagues' lab for multiple variables related to carbon, nitrogen, and phosphorus cycling and storage that we use as surrogates of ecosystem functions, together with estimates of productivity calculated using remote sensing approaches. So we are focusing on these variables because they are good proxies of nitrogen cycling, biological productivity, and also they are commonly used also as surrogates of supporting ecosystem services, which are important variables that at the end we are interested in and that can define overall ecosystem functioning. So this is really a very nice data set. We capture a really wide range of the variability that we can find in drylands today. We focus on natural, semi-natural areas. We didn't survey in urban areas heavily impacted by human activity areas. But other than that, we didn't control much about the land use. Many areas are used as grazing areas. Other areas are protected in national parks. But so this is some kind of a snapshot of how drylands look like today, or we have obtained data from most representative dryland ecosystems worldwide. So we have typical, for instance, African savannas, we have open eucalyptus forests from Australia, we have grassland and shrublands, and everything in between, and we have a relatively good coverage. These are just some examples of some of the sites that we survey. We have sites covering also a wide range of environmental conditions. Some sites are surveyed in Spain, the south of Spain, very close to the sea. Others in Peru, more than 4,000 meter height. Some sites have less than 100 millimeters of rainfall, like in the south of Tunisia. Some others have more than 1,000 millimeters of rainfall, like in Venezuela, in the savannah. So this is really a really nice survey. What we have been doing with this data that we are still processing, there are still papers coming out from this survey. I will just present some examples. And the first example I would like to show, this is one of the first papers that came out with this study over 6 years ago. And in many of our studies, we have been quite interested in exploring how changes in aridity across this spatial gradient, because we have a really nice gradient of aridity, from close to arid conditions, close to hyperarid conditions, all the way up to dry subhumid conditions. I didn't mention, but drylands, we are usually characterized by the ratio between the rainfall and the potential evapotranspiration they get. We usually follow the FAO aridity index, which is just this ratio, and drylands are all areas that have a aridity index below 0.65. So we are quite interested in looking at how aridity make things change in drylands, for multiple reasons. First, because aridity is like a major characteristic of drylands, and also because all climate change models forecast increases in aridity worldwide. No matter what happens with changes in rainfall, which as you know are much more difficult to predict because of global warming, aridity is increasing all over the world. So we have been using our survey to explore how things could change in the future in these areas following increases in aridity because of climate change. So in this study, we look at how changes in soil nitrogen stoichiometry, the contents of carbon, nitrogen, phosphorus, and the relationships among them in the soil across aridity gradient. Here we are using 1 minus aridity index to make the data more intuitive. So aridity increases from left to right. And what we found? We found that both organic carbon and total nitrogen follow this curvilinear relationship with important negative, important declines, linear declines, particularly in the transition between semi-arid and arid. But total phosphorus, the relationship was pretty weak in the case of phosphorus, but didn't follow the same trend. It slightly increased as aridity increased. If we look at the ratio between the different elements, I'm sorry, I need glasses. I cannot see very well from far away, so I need to get closer to the screen. So here is N2P, C2P, a C2N ratio. So when looking at this N2P and C2P, we follow the same curvilinear relationship that is not found when looking at the C2N ratio, which in this case we found with a very weak slope, but a slightly linear decline. So what this data suggests, that we found that as we increase in aridity, we found important declines in those elements that are mostly under biological control, carbon and nitrogen. And basically what we see is that you increase aridity, you reduce a lot vegetation, the cover of plants, and then the input, the organic inputs are reduced to the system. So carbon and nitrogen in the soil reduce, but phosphorus increases. So as a result, there is some kind of uncoupling. We get, even if you get an increase in phosphorus, you get a much more decrease in carbon and nitrogen. So the availability of this element is uncoupled, if I'm spelling properly. And this is very interesting because, as you know, the stoichiometry in the soil affects the stoichiometry on organisms, and this regulates many biological processes, physiological processes. And I'm not presenting results, but this is quite interesting because, as you know, and as you are studying here, phosphorus limitation is really a big worry in terrestrial ecosystems. But in drylands, it seems that we may not face the same problems, because even if we have more phosphorus available when aridity increases, our data also suggests, I'm not presenting them, that in order to make use of this phosphorus, as you know, the organisms need to produce enzymes like phosphatase, and the concentration of enzymes in the soil is strongly linked to the concentration of carbon and nitrogen. So even if we may get more phosphorus in the soil, as aridity increases, this may not be available to organisms. So all of this, I just wanted to show it first, just to put an example of something that we already know, is that drylands are ecosystems that are strongly driven by resources like water. So aridity is really a major driver of what happens in drylands. So is there any role for organisms to drive ecosystem processes, ecosystem functioning in drylands? And this is what we have been studying next, and I hope to convince you that this is really the case. This is the first paper that came out from this global survey more than 6 years ago. And here again we did a very simple study to look at a basic ecological question. What is the relationship between biodiversity and ecosystem functioning? As you know, this has been a question around for over the last 25, 30 years with a lot of interest by ecologists all over the world, and we use our data to explore these relationships. We are using this data for doing that. In the x-axis, I present the number of perennial plant species found in our sites. This is square root transform. And in the y-axis, we are looking at different multifunctionality indexes. This is also, multifunctionality is also a buzzword these days in ecology. And by multifunctionality, we want to characterize the capacity or the ability of ecosystems to provide multiple functions at the same time. So we value ecosystem because they do multiple things. They fix carbon, they provide with biomass production, they provide with soil fertility, they regulate water, regulate climate, they do multiple things, and they are doing that at the same time. So there has been a lot of interest in biodiversity ecosystem functioning research about moving from looking at particular functions like productivity to looking at multiple functions at the same time. This is what we call multifunctionality. In our case, in this particular study, we calculate multifunctionality in a very simple way. We collected our different soil variables related to carbon, nitrogen, and phosphorus cycling. We standardized them, and we made an average as our multifunctionality index. It's very simple to interpret. It has some issues, but for the type of data that we have, it works relatively well because we don't have trade-offs between functions. For instance, you can be in an ecosystem when having a lot of nitrogen is not good, because then nitrogen is leached, it can be going to the aquifers, or it can be denitrified, and then you increase greenhouse emissions. But this is not the case in drylands, where the more nitrogen you have, the nitrogen is very limited in the system. So we don't have trade-offs among our functions, so they are working relatively well for our data. So here I present results for multifunctionality, functions, carbon, nitrogen, and phosphorus cycling together. Function also only for nitrogen, carbon, or phosphorus cycling. In all the case, we found positive but very weak relationships. The scatter is substantial. So the first time I saw this, I was a little bit disappointed because it's the square of the models is really low. But when you think about it, it's not so bad, and it's not so surprising. Remember, this is an observational study worldwide. We are not controlling anything, and this is just telling you that you find a relationship that is still maintained once that you control by the spatial autocorrelation. And despite you get R-square models with R-square lower than 5%, as you see, getting a bad regression is not the end of the world. You still can get this stuff out. But this is telling you that there is a relationship, but it's relatively weak. So we are further exploring this data to see, okay, there is a relationship, but can we say that there is really a link? The biodiversity is important for ecosystem functioning or multifunctionality. So we did additional analysis with this data and did a multimodal analysis where apart from species richness, we include additional abiotic factors in the model that we know that are important predictors of dryland structure and functioning. We include climatic variables, we include soil variables like texture and slope, which are key drivers of the amount of water that gets infiltrated and stored in the soil, and we also control by the geographic for the autocorrelation, or data have autocorrelation, so we need to control for the coordinates, the location of the different sites. Sites within Spain are not as independent as sites between Spain and Argentina, for instance. So these models were much better. They explain more than 50% of the variation in the data, and we did here a very simple analysis. I don't know if you are familiar with this multimodal analysis, when you do all potential, multiple regressions with your independent variables, and you rank them according to the Akaike criterion. So the best model is the model with the lowest value of this index, and the most parsimonious model is the model with a similar value, usually with a difference lower than 2, and with the lowest number of parameters. So in our case, doing this analysis, we obtained more than 250 models. I present here only the best 8. All the models include the species richness, and when we include the best model and the most parsimonious model with the same models without the species richness, we got a really important decline in the Akaike information criterion. So this analysis suggests that even if species richness per se explains only a very tiny fraction of variation, is a relevant fraction that is not explained by other variables. We have been doing additional analysis to explore how different aspects of biodiversity are affecting ecosystem functioning. As you know, species richness, taxonomic diversity, is just one aspect of biodiversity. There is also a lot of interest about looking at the importance of things like phylogenetic diversity, functional diversity on ecosystem functioning. So we continue working on this, and in this study we look at the effects of both taxonomic and functional diversity as driver of multifunctionality. And here I'm not going to explain the statistics, which are a little bit complex. I'm trying here to summarize some modeling results where we include both abiotic and biotic predictors. In this case, the abiotic were variables like aridity, soil pH, sand content, also elevation and the coordinates. And we include species richness and different functional diversity metrics. We were able to we didn't measure this data in the field, but using TRI and other databases, we were able to reconstruct functional diversity for two important traits, plant height and a specific leaf area, for a very good number of sites. If I don't remember badly, I think we have functional diversity data at the community level for over 140 sites of the 236 in total that we survey. So the results of this analysis were quite interesting because the models were relatively good. They explain over 70% of the data, and the relative importance of biodiversity was quite high. They was almost as important as abiotic factors, and the effect of species richness was positive but weak, but the predictive capacity of the models improved a lot when we incorporated functional diversity. So including functional diversity was able not only to increase the predictive capacity of models, but also indicates that functional diversity is a much stronger predictor of multifunctionality than species richness per se. We are also doing additional analysis, but I just didn't want to include them not to put so much stuff. We are also have right now another paper under review on the effects of functional, phylogenetic and taxonomic diversity, which also quite interesting results. We have been also looking at the effects of taxonomic and functional diversity on other aspects of ecosystem functioning. For instance, in this study, we look at the impacts of these biotic attributes, also other abiotic variables, on ecosystem stability, which is also an important feature of ecosystems. In this case, we use remote sensing data obtained from the latest, from 2000 to 2016 in our sites, using MODIS sensors, and we look at the temporal stability in biomass production, in productivity. And we look at the drivers of this stability. And again, we obtain pretty good models, explain more than 70% of the variance of the data. And abiotic factors were important predictors. Of course, remember, we are the drylands. Things like aridity, soil features are important predictors of the variability. But again, species richness and functional diversity came up as important predictors. And in this case, we found an interesting interaction between aridity and richness. We explore this interaction further and look at how the things change between high sites with higher aridity conditions, sites with lower aridity conditions, and for doing this we use structural equation modeling. I think, are you familiar with these SEMs, right? This is an approach that we use a lot in our research because, as you know, they are very versatile in Spanish. How can I say that in English? Versatile, right? Okay, versatile. They are very versatile, and they are really very well suited for when dealing with multivariate data sets, observational data sets, where different independent variables are not fully independent among them. So you can model the relationship not only between your independent and dependent variable, but also between your independent variables. So these are very simple models where you include aridity, species richness, and different functional diversity attributes, in this case using mean height and the variance of SLAA. And what we found is in low aridity sites, the importance of functional diversity was much higher than that of species richness. But under high aridity conditions, the species richness became much more important predictor of stability than functional diversity. The results were quite interesting, not only because we are one of the very first obtained at the global scale using field data, but also because they suggest that in order to promote, the stability in biomass production, which is a very desirable property in ecosystems, particularly in areas that are many dryland areas that are being used for grazing, so we can try to manage by managing different aspects of the structure of the ecosystem, different aspects of the plant diversity in these ecosystems. We have been also working using this database with microbes. We were able to get frozen samples, soil samples. As you can imagine, the logistics involved in these surveys are challenging. you are in the middle of Botswana or in the middle of Patagonia, you don't have a freezer nearby, so it's not easy to process the samples or particularly to obtain frozen soil samples for doing molecular work. But we were able to manage to get these samples for 80 of the sites, and we are also now looking at soil microbial communities using DNA techniques. This is the first paper we came out with this data. It came out just 3 years ago. And again, a very simple study. We look at the effects of aridity, in this case in the diversity and abundance of both bacteria, soil bacteria and fungi. We quantify them using DNA approaches, Illumina for diversity, for sequencing, and qPCR for quantifying the abundance. In all the cases, we found a relatively clear pattern, a linear decline, a quite strong decline in both the abundance and richness of these microorganisms as aridity increases. So we further explore this pattern using, again, SEMs, structural equation models, where we include also other predictors other than aridity that we know that are important drivers of soil microbes. We include, for instance, soil organic carbon, pH, the cover of plants, and also variations in mean diurnal temperature range, variation in diurnal temperature, which are quite important drivers of bacterial activity in many natural ecosystems. And we have explored the effects of these predictors on both the abundance and diversity of bacteria and fungi. In both cases, for both sets of organisms, what was really interesting is that we didn't find a direct effect of aridity on the abundance and diversity of soil microbes. So these relationships that we found are really driven by indirect effects of aridity. This is also a very nice thing about SEMs, that you can disentangle direct from indirect effects of your predictors on the variable that you are modeling. Aridity didn't have a direct effect, as I'm saying, but it really had strong effects on variables like soil organic carbon, on variables like plant cover, and variables like soil pH. And organic carbon in particular were strongly positively related to both bacteria and fungi, which is not a surprise. We are dealing mostly with heterotrophic organisms. They need carbon, right? And their main inputs, they usually come from plants. So as I said before, you increase aridity, you reduce plant cover. This has a negative effect on the amount of organic carbon, which is strongly positively related to both the abundance and diversity of bacteria and fungi. And with this, in this graph, I just showing the standardized effects of the different predictors in the model, and you can see clear the importance of organic carbon, particularly for bacteria. We also did additional analysis using this data to look at the impacts of how microbial diversity affect ecosystem multifunctionality, as we have done previously with plants. Again, we use SEM. We include predictors, important predictors of both microbial diversity and multifunctionality. And what we found is a very strong positive and direct effect of microbial diversity on ecosystem functioning. As you know, those of you that are familiar with SEMs, once that you see a direct path, these direct effects already take into account the other effects that you include in the model. So you can assume that is an effect of this variable on the other. But remember, we are dealing with observational data. We never can be sure that this model is right. The SEM what tells you is you have an a priori model, as you know, and you fit it to your data, and you get a good fit to your data. That means that your data fit the model. That doesn't mean that the model is right, and that doesn't mean that this is the best model that you can get. Remember, this is observational data. We never can be sure about cause and effect relationships using this type of survey. This is something that I always like to remark, because it's a common critique that we obtain from this type of work, which of course I assume is not a critique of our work, it's a critique of this type of data, this type of observational studies. But still, we found evidence, or according to these models, that microbial diversity has a direct effect on ecosystem multifunctionality. And when we look at the total effects of the different predictors, not a surprise, for instance, things like annual rainfall has a positive effect on multifunctionality, not a surprise. We're in drylands. The more rainfall you get, the more biological activity. Ecosystems function at a higher rate. You increase temperature, you get a very strong negative effect, not a surprise. And what is interesting to see is that on the one hand, the magnitude of the effect of microbial diversity was pretty high, and within microbial diversity, the magnitude of the effect of fungi was much higher than that of the effect of bacteria, which is quite interesting, and this is something that we are also exploring right now. So we have seen that plant species richness increase multifunctionality. We have seen that microbial diversity increase multifunctionality. What happens when you put both in the same model? We did this, and in this model, very similar as the previous one, a little bit more complex. And the first thing I would like to emphasize is that this didn't link that we have observed of this relationship between richness, plant species richness, and multifunctionality didn't become evident. It was not significant. Is this a contradiction with our results, previous results? Not really, but because we found a strong effect of plant species richness on microbial diversity, which is strongly related to ecosystem multifunctionality. So what this is telling us is that the effects of plant species diversity that we are observing are mostly indirect effects, which are driven by how plants affect microbial communities, which is not a surprise, because here in this study we include plant productivity as one of the functions, but most of the functions that we evaluate are related to carbon, nitrogen, and phosphorus cycling in the soil, which ultimately are driven by soil microbes. So it's not such a surprise. We are also using, and I'm getting close to finish with this part, we are also using data and samples from this global survey to do incubations and experiments in the lab under controlled conditions. So for instance, in this study, this is a very interesting study that Manu Delgado did. And here what we did is we collected soils from our survey, from these 80 sites where we characterize microbial communities. We incubated them in the lab under simulating different climate change or global change scenarios. We simulated them under different temperature conditions, we simulated them under different nitrogen deposition conditions, and we incubated them simulating drought, different drought intensities. And the objective of this particular study was to look at the, it's a buzzword, resistance of multifunctionality to global change drivers. Okay. Multifunctionality, we characterize that with different functions as done before. And I don't know, I guess you are familiar with the resistance and resilience concepts, right? These classical ecological concepts. Resistance is if you disturb something, how resistant is the system to move, and resilience is how long it takes to get back to the normal conditions when the perturbance is gone. So in this particular study, we look at the resistance of multifunctionality to these different global change, simulated global change drivers. And in particular, we were particularly interested in looking at how the composition of microbial communities affect this resistance to global change drivers using an incubation experiment. And again, we use structural equation models, and we found a quite strong effect of the composition of microbial communities on the resistance of multifunctionality to global change. And in particular, there were some particular species, not a species, because we are dealing with OTUs, and this technology still is not very well to characterize species, but some particular groups of bacteria and fungi were either very strongly negatively related or strongly positively related to the resistance to multifunctionality. And this is quite interesting because it gives us information about which organisms not only can be winner or loser, but which organisms, if we are at some point are able to manipulate soil microbial communities, can help us to make our systems more resistant to these to the consequences or to global change drivers. This is an incubation experiment. We also have been doing incubation experiments to look at topics that also are receiving a lot of, have received and are receiving a lot of attention. For instance, to biotic feedbacks to soil respiration and to climate change. As you know, there is a huge interest on trying to understand not only the carbon cycling, but how key aspects of the carbon cycle, like soil respiration, are responding to ongoing climate change, because this really can make a huge difference in the global warming of the planet. And this is really a hot topic right now, in trying to understand how we can understand how this works in order to make better forecasts, better predictions. And one of the key topics that are being studied with this large topic, is what is the role of microbial communities under warming. There are multiple studies suggesting that some microbial communities can adapt to warming. If this happens, then as warming goes up, microbial respiration will go down or become stable. But other studies are showing the opposite. That is, in many ecosystems, microbial communities are limited by temperature, so you warm up the system, they respire more, they release more carbon, and this will have a positive feedback with climate change. So we have been, and as you can imagine, most of the studies have been carried out in ecosystems other than drylands, so there is a lot of uncertainties about how soil respiration is going to respond to climate change in drylands. And also something quite interesting is when you look at this literature, which is becoming larger because there is a lot of people working on this topic and these issues. Most of this, the response of microbial communities or the soil microbial driven soil respiration to climate change has been carried out either using short-term incubations. People take soils from one place, take them to the lab and make short-term incubations, or using climate change experiments, like the experiments, for instance, that Josep and his group are doing at CREAF, or some of the experiments, like I showed, that we are doing in our field sites. Either using warming devices, you warm up for some years, and then you look at how things change over time. But these are still, very few and very limited in time. And when you look at this type of studies, for instance, that have been done with plants or with animals, they are usually done like at the microecological scale, looking at these really large scale gradients. So we use something quite interesting from our survey. When we start to look the data that we have, our survey really encompasses, really captures a really wide range of mean annual temperature conditions. We have some sites in China, we have a mean annual, are really cold. Remember, drylands, we have always the image of hot drylands, places that are very hot, but there are very cold drylands. Antarctica is a dryland. It's a cold dryland, but it's a dryland. And for instance, we have sites in China with a mean annual temperature lower than 0, and we have sites with mean annual temperature close to 30 degrees. So we have really nice gradient, and we have been doing incubations in the lab to look at how respiration changes and what is the role of microbial communities on this process. So this is a paper just came out. Just became accepted. So you are one of the few on looking at these results. So here we do the incubation in the lab under excess of substrate, so without the substrate limitation. We are also controlled by microbial biomass using different approaches, because this is very important. As you know, respiration is dependent on microbial biomass, and you cannot kill the microbes, because if not, there is no respiration. So we control using qPCR, using the substrate SCR, substrate induced respiration method, and using also chloroform extraction to quantify it at the end of the experiment. We control microbial biomass, we control substrate, we only modify temperature, and here I present potential respiration. These are not actual measurements, are respiration ones that you control for all these factors, so that's why you have these clean lines. And something that we observed, and we did this experiment incubated at different temperatures in the lab. So as you can imagine, you increase temperature, you increase respiration. This is not a surprise, because there is no limitations. And I didn't mention, but there is also no water limitations. We put them 80% water holding capacity, so the microbes will have plenty of food, plenty of water. So they were not limited by these resources. And something that we found that was really, really interesting is, no matter at which temperature you incubate your soils, you always find this negative relationship between mean annual temperature and potential respiration. That means, or that suggests, that microbial communities are adapted to the mean annual temperature conditions of the site, which is something that it has not been shown before using this type of empirical data. And also because we collected soil samples from, I didn't mention before, but in our survey we collected soil samples under the canopy of vegetation and soil samples in areas between plant patches. We were able to test small scale differences in resource availability. As you know, in drylands, under the canopy of plants, you have more resources. You have more nitrogen, you have more carbon, you have higher biological activity, higher microbial activity. And again, we found the same trend, but locally, respiration is increased under the canopy of vegetation, which also makes sense. So these results are very interesting, because it's the first empirical evidence suggesting that microbial communities are adapted to local temperature, in this case, mean annual temperature, and this suggests that if you increase temperature with warming, soil respiration, at least in these drylands, may not increase, because communities, and as you can imagine, these organisms at the end, they have physiological, like organisms, we face similar physiological trade-offs. If you are in an area which is really dry, you really need to adapt to these conditions to resist these strong aridity conditions. So even when you get fluxes of water, you cannot function at a higher rate because the microbial communities there are not really adapted to function at a higher rate, to respire a lot, to say in other words. So this is just some of the examples that we have done with this global data. We have done many more, but of course I just don't have the time, and it will be very boring to start, talking a lot about this. And just getting close to finish, but just to show some of the local scale studies that we are doing, some of the experimental work we are doing. Because, as I mentioned before, one of the critiques that we get a lot in our papers, we get a lot of rejections, and many reviewers are very critical. There is no cause and effect relationship using this data. You cannot really prove what you are saying. I say, of course, come on, I know. But if you can do an experiment in more than 200 sites all over the world, keeping them for 10 years, go and do it. Go and do it. And that will be fantastic because it will be much better than this. But these are the best data that we have at the moment to do this type of work. But we are also combining this data with manipulative experiments, both in the lab and in the field, to try to see if we can validate experimentally some of the patterns that we are observing at the global scale. And for instance, we are doing experiments using our Biological biological soil crust, which are these communities that I personally love. I really like working with these guys. They are very, very interesting, really challenging because the taxonomy is complicated. They grow slowly, they are not plants, but they are really, really, really important in drylands because, as you can see from this picture, this is one of our study sites in the south of Madrid. All these white things here, perennial vegetation cover is just less than 30% of the surface of the landscape that you can find in these areas. Most of the rest of the area is covered by these communities. So they are really, really important because they modulate lots of processes. They modulate how water infiltrates. They modulate how seeds germinate. They fix carbon. They fix nitrogen. They have particular microbial communities associated to particular lichens. They produce a lot of secondary compounds. So they are really, really, really important players in this system. So if you want to understand how drylands function, how they work, you really need to understand what these guys are doing. And also this, because they are small, they are difficult to work with because of the taxonomy, but they are small. So this has an advantage that you can do, climate change experiments. I would like to say, the Spanish way, without many resources, without a lot of imagination. So we can, I have seen the things that you have in the garage. This is fantastic, but we just don't have the logistics nor the money to do this in many of our sites. So we are using this system, that is the system that people use in the tundra, for instance, these open-top chambers, to simulate global warming. And we are using these shelters, this type of shelters were originally proposed by Osvaldo Sala and his group in Argentina, back in Argentina, when they face the same logistic issues as we face, and they are working really well. So we have now some of these experiments. We have 3 experiments like this, one in the south of Madrid, another in Alicante, another in Almeria. And some of these experiments, the one is running for 10 years, the other for 9 years, and the other for 8 years. Depending on the money that we get at the time, we were able to set up. And we are really observing really very interesting results, because with these experiments, we are not only interested in looking at, how climate change is affecting these communities and the process that depend on them, and multiple ecosystem processes, but we are also particularly interested in looking at how the attributes of these communities, the composition of the species composition, diversity, the spatial pattern, the abundance, how these communities and their attributes modulate ecosystem responses to climate change. So for instance, some of the results, this is from the site we have in Alicante. This paper just came out recently. We are here summarize the results of the first 8 years of the experiment. And for instance, we found with warming, this is for areas that initially have a low biocrust cover, areas that had initially a high biocrust cover. In these areas from the very beginning, with warming, we saw a decline in soil respiration, which is what we are representing in this graph. In the areas with low biocrust cover, this didn't become apparent until several years later, but again, the long term also soil respiration was reduced with warming. And we collected soil samples and did multiple analyses pointing, for instance, we found a decline in the respiration per unit of microbial biomass. We found declines in the production of, for instance, enzymes like beta-glucosidase. So we found multiple lines of evidence suggesting that microbial communities are becoming adapted to this increase in temperature. With this matches pretty well the results that I showed before obtaining with the global survey. We also are doing experiments of this kind with plants. We set up a really nice facility at the university, Region College University. We have this common garden facility with a lot of open top chambers, more than 100, and so 100 something I think, and multiple rainfall shelters. So we can do complex. We have been doing multiple really highly replicated and complex experiments using biocrust, using plants, also using microbes. And for instance, in some of the experiments that we have done with plants, with these experiments, we did an experiment crossing multiple species richness with climate change drivers in an experimental way. So the idea was to see, how the direct and indirect effects of climate change on ecosystem functioning, driven by changes in species composition, richness, functional diversity, and so on. So the type of things that we have been looking at with the global database, just setting up experiments to try to disentangle experimentally what is the role of the different factors as drivers of changes in ecosystem multifunctionality measured using the same set of variables that we have using in our global survey. For instance, what we found here, this is a variation of structural equation models. It's not a structural equation model, it's another statistical approach, but it's trying to, the representation can be interpreted in a similar way. There are lots of arrows, small numbers. I'm not going to enter into the details, but in this particular case, we didn't find a direct effect, a strong effect of climate change drivers. We found a strong effect of species richness, and the effect of climate change drivers on multifunctionality were mostly driven by the changes in functional diversity promoted by climate change. In our experiment, climate change, particularly warming, promote larger plants. Larger plants increase bacterial abundance a lot and also increase the abundance of protists, and these modifications led to a higher multifunctionality. So experiments also provide us with the capacity to become more look at particular mechanisms and interactions, but again these results point out at the importance of the attributes of communities like species richness or functional diversity as drivers of ecosystem functioning multifunctionality, and that, as we have also getting evidence, many of the impacts of climate change will be indirect and driven by changes in the biotic communities that they induce. And I'm getting close to finish. Just 2 minutes more and I will be done. So what's next? So we are still continue processing data from these experiments are still ongoing. We are still getting out data and analysis and papers from our global survey. And that's interesting because this global survey that I have been presenting, everything started, as I like to say, with one table, one computer, and 40,000 euros, which is what I got when I got a Ramonica Hall in 2005. This was my starting package. People don't believe me. This is the starting packages that we get in Spain. If you are lucky, an office. If you are more lucky, the university will provide a computer with a desk, of course. And if you are lucky and get a Ramonica Hall, now it's better. You get 40,000, but at the time I just got 14,000. So anyway, we started getting small projects. We did a gradient in Spain, Spanish sites that worked pretty well. And then we had the chance to look for one of these CITED networks. I don't know if you are familiar with CITED. This is an organism that found networks, research networks between Spain, Portugal, and Latin America. They don't give you money for doing actual research, but they give you money for creating a network, gather meetings, and so on. So we got one of these networks, and what we asked our colleagues in Latin America was to replicate what we did in Spain in different Latin American countries, which worked pretty well. And then I got an ERC starting grant, and then the things become global. But it was not something to be global from the very beginning. Everything started with the money of the Ramonica Hall and a gradient in Spain. But when I had the chance to apply for a consolidator grant, then I designed another global survey to be truly global from the very beginning. And in this case, what we are looking in this new global survey that we are doing right now, is getting close to finish, is we are looking at the interplay between aridity and grazing. Because aridity, as I have tried to show, is a key driver and is a proxy of climate change in drylands. And also grazing is very important for multiple reasons. First, because it's the main land use in drylands all over the world, not in places like Spain or Europe, but if you go to Africa, you go to South America, large parts of the US, large parts of Australia, the main land use in drylands is not agriculture, they are not very productive, but is grazing. Grazing is really important. And it's not only it's a very important land use, but it's a major driver of land degradation and desertification in these areas. So in this consolidator grant, we are looking at the interplay between climate change and desertification, and one of the key things that we are doing is this global survey, where in every site that we are surveying, we are getting data across grazing gradients. So what we have is, for instance, this is 3 sites we have in Spain. Within each site, we have 3 actual plots in the field with different grazing intensities. So the idea is that for every aridity level, for every place where aridity and soil conditions, vegetation is the same, the only things that will change is the intensity of grazing. And we are looking how this impacts ecosystem structure and functioning using the same set of variables I have presented before. This survey is working really well. We are getting close to finish. Right now we have already data for over 330 plots over 300 plots in 25 countries have been already surveyed. And if everything works as planned, we'll have over 330 in 27 countries. So this is really, and this time, for instance, we are measuring everything that we have measured in the past, and we are measuring, for instance, new things like plant functional types in the field. We have already processed data for over, I think the numbers are really high, over 20,000 individuals that we have measured so far. And we are going to also measure isotopes and a bunch of things with the objective to look at the interplay between aridity and grazing as 2 major global change drivers and desertification drivers in drylands. We have been also using our dryland data. We have also putting this data together with all the data from other ecosystems to look at more global responses. This is an example that came out this year, again, work by Manu Delgado. He combined data from our survey with data from other surveys to look at changes in, just to look at microbial communities. And we really found something really interesting is that a very tiny fraction of phylotypes, if you look at all this is sequence data. When you look at the sequence across the board, across the world, this use data from over 240 sites from all sorts of ecosystems. So only just less than 2%, over 2% of the reads of the phylotypes accounts for over 40% of the total number of reads. So this is a very few taxa that are really dominant in soils all over the world, and we also use this information with, we did different analysis to look at which environmental drivers drive the abundance of these abundant taxa, and we were able to create these global maps of dominant soil bacteria, which became really, it's a very interesting study. We are doing exactly the same with fungi. This paper is right now under review, and we found very, very similar results. In this case, less than 100% of the fungal phylotypes account for over 20% of the fungal communities in soils from all over the world. And we also here in this case are looking at the genes that are for those genomes that are available, which are the genes that makes this species, or which are the traits that make this species so successful. And we have, for instance, observed that these species have a higher frequency of genes related to dispersal, related to stress resistance, which is what makes sense, no? Species that can disperse easily, a species that can I know how it writes, withstand, aguantar in Spanish. Species that can thrive over a wide range of environmental conditions are those that are able to colonize many sites. So, and this is also very, very interesting work. And that's it. As you can imagine, I'm here alone talking, but as you can imagine, all this work is the result from the efforts and work of a lot of people. And so I would like to thank particularly everyone in my lab, that as you can see here is a quite young and large and dynamic group of people, all our collaborators all over the world, of course, the funding agencies that have been funding our research over the years, of the ministry, the Fundación BBVA, the British Ecological Society, my university, Comunidad de Madrid, and of course now the ERC that has been funding what we have been done over the last 8 years so far. So this is really nice. And of course, thanks to you for your attention, and if you have any questions, I will be more than happy to try to answer them. Thanks.
Thank you very much. It's been very fascinating. So, questions? Pilar? I have a deal of questions.
Thank you very much.
You're very welcome.
I was thinking just now, I am starting to prepare a project in your Aridlands in Alicante with Andreu, and we have a deal of methodological constraints. You have mentioned some of them. So one is the heterogeneity associated to aridity, but another one you have just mentioned now is that most of our areas, those are abandoned. This is the old chronosequences of post-abandonment. Mm-hmm. We are retaking this for soil and vegetation over the cross sequences. So we have grazing there. So I was thinking about what I discovered in the States, in the Great Plains, is that the kind of animal, this is not only the intensity of grazing, but particularly the species.
It's crucial.
That's terrible, because sometimes there is no influence of big mammals because the system is already has co-evolved with the big mammals, but we have the small mammals that are more influential. So how are you introducing this variety of animals?
Yeah, we are, of course, as you said, this is a very important issue. It's not the same, a goat than a cow. And it's not the same if you have rabbits or if you don't have. And it's not the same if the system has evolved with grazing for, 100,000 of years, or systems that, people now are putting animals there. So we are controlling this. It's not easy to control. It's really difficult, because even in areas, you can go to Argentina that, you can go to Patagonia, where it's a huge issue, because this is what people make a living of, right? It's just raising sheep. And they have, very well controlled. And you go there, you try to quantify, how many animals you have. I say, well, I have this potrero, which is what they call in Argentina the areas people manage, haciendas or fincas. But a potrero can be, 10,000 hectares. So, no, the density of animals in this area is this one, yeah, but then you go to the field and it's impossible to know exactly the density of animals in the particular area. So what we are doing is we are quantifying them indirectly. So we are doing, of course, I didn't have present any details, but we are measuring the number of, and of course we are first gathering data with the managers and with the local people. In some areas that we have very well characterized, for instance in Madrid, we have an area which is just nearby a shepherd. So, we know how many animals, we talk with him, the routes that he takes with the animals and so on. But no matter the information that we can gather of this kind, we quantify in the field indirectly the intensity of animals and the type of animals using the dungs or the pellets. This is very well correlated with the density of animals. And of course, just by looking at the dungs, if you have cows, you have rabbits, you have goats. The people working with this, they know how to, well, of course, I can differentiate the dung of a cow from the pellets of a rabbit, but if you put the pellets of a rabbit and the pellets of a goat, they can also be differentiated. So we measure them, we take them subset to the lab, we weigh them, and there are lots of studies at the local scale showing a very good relationship between the number of pellets and the density of grazer.
Thank you. We're going to take.
And also, it's very important to take into account if you have natural grazers or not, because you go to Australia, then you have kangaroos. You go to Argentina, you have vicuñas. You have vicuñas, so that there are rabbits here. There are rabbits, so this is important to take into account.
Thank you.
Yeah, welcome.
More questions, please.
As you are talking of grazing and dams, you are taking into account the effect of the dams in the fertility of the soil and the positive effect of this rating?
Not really. We are not really specifically looking at this. First, because the complexity of the logistics involved in this type of survey are challenging. So this incorporates like another layer of complexity that is not easy to handle, because then you enter into issues about seasonality, because depending on when the animals are in, and if the dung is fresh or not, the effects of on the natural cycling, they vary a lot. But we are not. Of course, we will get data on the incentive, the number of dungs worldwide, so we can try to correlate them with the different variables that we measure, but this is not really a factor that we are looking at. It's really beyond our capacities, and also it's beyond of the scope of this survey. But yeah, it's very interesting. It's really interesting.
Well, first, thanks for your exciting presentation.
Yeah,
That's
very welcome.
Interesting. My question is more related with the soil fauna, because we are very interested in analyzing the effects of soil fauna communities in terms of organic material decomposition and nutrient cycling, and I miss that they are not included in the work that you present. I don't know if you have studied them in your site, and or if you are planning to include them.
Yeah, we have not done anything with soil fauna in the past. We are starting now because in the past we have using molecular work. ITS and 16S, which is mostly for fungi and bacteria, but now we are going to use 18S, which is another primer for more general primer for eukaryotes. So the idea was and we have been also working with particular primers for protists, but we have not worked so far with things like microarthropods and so on. Now we plan to characterize them by using molecular approaches. As I say, because of first because of lack of expertise, we don't have any expertise on soil fauna, but we are, for instance, now collaborating with groups in Germany, the group of Nico Schenauer, Michael Bonkowski, that they have long tradition working with earthworms, working with protists and other soil. We are working with Matthias Willig also in Berlin, that he's also working with microarrays and so on. But we are strongly limited by, spatio-temporal constraints and logistics. So everything that we will do with this, that we plan to do it, but we'll do only using molecular approaches. It will be great, having actual surveys about, which type of insects or microarthropods, just looking at the things like termites, which are very important, all over these systems. But it's really very difficult to do.
Thank you. I thought it was very interesting, your pattern of soil respiration with temperature, that as temperature goes up, the average respiration goes down. I was wondering if in these dry soils, when there's a rainfall pulse and respiration goes up, does that peak also change with the temperature?
We have not specifically looking at it. We know that it happens. There is a pulse of water, there is a flux of respiration, but we have not specifically looked at. But I will bet that this will happen too, because at the end, this is a physiological constraint, right? Of course, if you put water, they respire more, but they cannot respire more as they can. Let's say, if you can run 20 kilometers an hour, you can put a lot of gasoline, but you will never go faster, right? And the evidence that we are gathering is that these communities, let's say, if you are able to survive under very extreme conditions, you cannot be very active. And we have I don't know, have you seen this very recent study, beautiful study done in Atacama Desert in Chile, that they had this recent flooding, and this has wiped out the microbial communities, just eliminated them, because you have these communities that have evolved, over hundreds, thousands of years to almost no water, and all of a sudden, in 1 year, you get a lot of rainfall, and they have disappeared. So we have not specifically looked at this. We are now doing additional studies according to this, after these results, but my personal guess and bet is that this pattern will be also reproduced, because at the end is that you have a particular microbial community adapted to these conditions. So let's say this is like running a marathon. It's a trade-off between running fast and running for more kilometers, right? If you are ready to run for a lot of kilometers, you cannot be really fast. So it's this type of trade-offs is what we are observing or what our data are suggesting, and I think that this should reproduce also when looking at the impact of pulses on respiration.
Thank you.
You're welcome. Jordi.
Thanks very much for this impressive talk.
You're very welcome.
I have one question that goes similar. I found very intriguing these results of the soil respiration and temperature. And I understand the concept. These communities, soil communities, are adapted to their conditions. They are not able to increase the respiration with temperature, which is what a very naive model would predict. But at the same time, your data, if I understood it correctly, shows very clearly that in the short term, they are able to respire much higher rates. So I'm just curious to know exactly how you see these trade-offs that are involved with this longer-term response that really keeps the respiration down. If you have an idea of exactly what they are.
This is, as you can imagine, this is like the key issue, because I didn't mention this incubation only lasted for 10 hours. And the reason for last for 10 hours was to avoid, because one of the problems of long-term incubations is that they become adapted to the adaptation. So, so it's really very tricky. So this is really like the key issue. The problem is that, it's not the problem, it's the way it is in many fields or many ecological topics, is that, most of the studies are done for example, there's a lot of interest in the Arctic. So in the Arctic, if you warm up, they are going to respire a lot because, temperature is really a strong, it limits a lot the physiological activity of these organisms. But as we see, you are in the middle of Tunisia, temperature is not limiting, it's water. And of course, these communities have not evolved under, they have not evolved under plenty of water. They have evolved under a lack of water and plenty of temperature. And if we believe what is going to happen, we are going to have less and less water availability, more aridity, and increasing temperature. So we really cannot use the data and models from other ecosystems to drylands because they work in a different way. Of course, these are not definitive. This is just the beginning. We are just starting to explore these issues, and there are many things that we don't know, particularly how well these short-term incubations can really represent what happens at the long term. Because at the end, the idea is aridity is going to increase, communities will evolve. Some microbial species win, some will lose. But even if, can you imagine that more efficient communities came up, which I really doubt, even if they are more efficient, you are going to have less water, so you are going to have less respiration. So I think at least for drylands, this pattern to me makes a lot of sense. Of course, there is still a lot of uncertainties, and I don't want to say that this is the truth and that you need to believe this, and this is what is going to happen, because we are just starting to scratch the surface. But really what this suggests is that drylands don't work as many other systems have done. And this is very interesting because this paper will come out just at the hand by hand with another paper, because this is from a PhD student, Marina Dacal. So I sent her to Mark Bradford. Mark Bradford, he's a colleague working in Yale. He's an expert on soil respiration. He's a friend of mine because we were postdocs together at Duke. So I sent Marina to Mark because I know that he's an expert on this. And then when she arrived, they were doing exactly something very similar as we wanted to. We did, but in the field, in LTRs in the US, from Alaska to Puerto Rico, on over 2 years in the field, and they observed exactly the same pattern. And Mark had the idea, which this is only something that the Americans can do. Okay, this is really cool. Both papers, reinforce each other because two different systems, two different setups done in a very independent way, they go to the same direction. So we submit the papers together to the journal, they will review together, and hopefully, and luckily, they have been accepted together, so they will come up together. I will never imagine, approach to the editor of the journal and say, look, would you be interested in 2 for the price of 1? Can you review 2 papers together? And yeah, they like it, and so I don't want to say that this is the truth, but it seems that this may work this way. Let's say Alaska is not as New Mexico, and the drylands in particular are quite different to other types of ecosystems because, current drylands have not only evolved under current climatic conditions for a long time, but if you go farther down or deep in time, not millions of years, but you go thousands of years back, many of the areas were drier in the past than today. So these communities have been evolved for thousands of years under very dry conditions. So even, of course, you put them water, of course, you know, they are organisms, no? They need water, they function, but there are limits. You know, I like to put examples of the marathon, because unless you are a, you know, one of these African super runners, you know, you cannot run a lot if you want to run 42 kilometers, right? Because if not, you will collapse before the finish. So, but yeah, it's really interesting, you know.
And I have another very short question. At the very beginning, you presented these results on the effects of species richness, diversity, and there was a slide showing a very strong effect of skewness, of distribution of trait values, I suppose. How is that? Why is
this is very interesting. Well, it's a little bit complex, and I have to say that I don't fully understand it. I understand it, but it took me months to understand it. This is the work of two postdocs, two French, crazy French. You know, you work with the French, but they are crazy about these functional traits. And when they came to the lab, they had this idea, because most functional, you are familiar with the functional diversity literature, you know, you know, any variable, any distribution have 4 moments: the mean, the variance, the skewness, and the kurtosis. And most of the functional diversity metrics developed so far, they only use either the mean, when you talk about the impacts of the abundance, no? the community weighted mean metrics, they take into account that the dominant species, and functional diversity, they look at the variance, you know, the variation in the traits in your communities. But they have ignored the skewness and the kurtosis. And they provide very interesting information, because let's say, if you can link the skewness and the kurtosis to the concept of, for instance, niche filling, right? Let's say if you have a very skewed distribution, it means that, like, you know, some particular traits are very abundant and some others are like non-abundant. But this is not good from, let's say, if you want to fill all the niches, you want to maximize, right? This, let's say, minimize, have a less skewed distribution. And with the kurtosis is something similar, right? A highly kurtoki, como leptocurtica, y bueno, anyway, I'm sorry, I cannot tell this in English. If you have a distribution like this, you know, with a very high kurtosis, that means that you are like filling only like a small fraction of your trait space. So what these guys came up with this idea for using the 4 moments, and it's not only that, is that they came up with a massive, it was crazy when I saw this, I said, oh my goodness, they started to explore the data, and there is a mathematical relationship between skewness and kurtosis. It's a mathematical, you know, it's just pure maths. So we went with some physicists and mathematicians, and they help us to solve the equations. And on the one hand, there is a mathematical relationships between the skewness and the kurtosis, and then we discovered that this relationship is being used in multiple fields, in astronomy, in microeconomics, in all things you can imagine, to maximize functions. Let's say, for instance, this is something, well, I don't want to get on the branches, but for instance, this is people that use, when dealing with a national budget or a government budget, and you have a bunch of competing departments for the money, there are some, this equation is used to try to maximize. So anyway, the idea is that with the skewness and the kurtosis, you can model the theoretical maximal functional diversity you might get at a site. And that's something that you know, if you go to the paper, this is like one part. The other part of the paper is all the modeling, and it's a model that predicts, or that our data suggests that functional diversity is maximized locally. No matter the resources you have, functional diversity is maximized locally. And this maximization is driven by this relationship between the skewness and the kurtosis of the traits, which I don't know if I think I'm getting out for the branches, and I'm not sure if I'm responding to your question. But the idea is that by looking at the mean, the variance, the skewnesses, and the kurtosis, you can get like a more complete picture of how this, let's say, the functional niche space is occupied by your communities. And also a nice thing about using skewnesses and kurtosis as indicators or not proxies, as a metrics of functional diversity, is that, you know, the mean and the variance are strongly related. Let's say, or sorry, not strongly related, no, I made a mistake. Let's say many indices of functional diversity based on the variance are not independent of species richness. Let's say usually the more species you have, the more functional diversity you have. But the skewness and kurtosis are pretty much independent. So you can get like information about the pure effect or the independent effects of functional diversity using the skewness and the variance. No, sorry. The skewness and the kurtosis. This is really interesting stuff. We are now working on the mathematical properties of these relationships and doing some modeling. And we are getting, because we are reproducing these patterns that we have seen in our drylands with, for instance, sites in Germany, tropical forest data sets. So it seems that it could be something universal. And we are doing now some mathematical model to see if we can come up with some general or try to understand this. It's really interesting. It's really interesting.
Well, one thing that maybe I got lost, but you are doing this relationship between skewness and kurtosis for single traits or for
no, for single traits. For single traits.
So when you are speaking about the filling of the niche, how do you implement this with?
Yeah, for instance, we are talking, for instance, no, yeah, we are only, for instance, with volume or height, right? Let's say, if you have a very skewed distribution, that means that you only have like one size, right? And of course, it depends, because if you only have tall plants, that's good. For instance, because tall plants, you know, they have more, they are larger, they usually fix more carbon, have more biomass. But if this is just the other part of the your skewness, the other way, the other part of distribution, that's not good. And if you have like a more even distribution, no, which at the end, then that means that you can fill better your space, because that means that you have tall plants, small plants, you have and everything in between. So this is the idea. With the specific leaf area is the same. Let's say if you have, for instance, that happens in drylands a lot. If you had this, as you know better than me, no, you have been working on these issues also for a long time. When you have these very recalcitrant leaves, you know, they are not very good for the soil in the sense that they take a while to decompose. They are not very nutritious for animals and so on. And so if you have like a much more even distribution in your specific leaf area values, that's good for ecosystem functioning, because that means that, you know, you have a higher decomposition. Because in dryland, the scenario, the opposite doesn't happen. You know, dryland species, they don't have very dusty leaves that can be decomposed easily, you know, because there is no water, they cannot maintain this type of leaves. But yeah, we have not done anything at the multivariate space yet. And one of the reasons is because we didn't have the data. We are now starting to having data, so maybe in a few years from now we will be doing this with a multi-trait space. But right now, this is everything we can do.
So let's leave it like it is now, and thank you for your...
Thanks a lot, yeah, for coming, your questions and the invitation.
The reflections about these mathematical issues that maybe we need to contract on mathematical.
Yeah.
It's further than...
It's really further. It's beyond, many of these things are beyond my understanding too, but it's really interesting. When the people that knows explain in relatively simple terms, and what is really important is because at the end, these are patterns with particular data, but if you can find like mathematical models following general properties that match your data and other data, then that's the key point, because that means that what you are observing is part of a much broader general pattern. Yeah, this is, we don't get bored, you know.
Okay, thank you.
Thanks.